Thank you, everyone from around the world. My name is Lorraine Rush, Vice President of Scientific Strategy and Commercial Development at Celerion. I'd like to go over some housekeeping items for today. The estimated time is 40 minutes for our presentation, plus time for Q&A. Remember to submit questions via the Q&A button, and we will address as many questions as time permits. Any questions that we don't have a chance to answer, someone from Celerion will address directly within 24 to 48 hours. I'm very excited to share the new developments around our high-res mass spec expansions at Celerion. Per our recent press release, this capability allows our clients to work with us from in vitro through in vivo metabolite profiling, spanning the non-clinical and clinical development phases in accordance with the appropriate FDA and EU guidances pertaining to the identification of major metabolites as part of a regulatory submission for product approval. Today, we are fortunate to have both Fred Pritchard and Sergio Menta sharing their regulatory and scientific expertise to help position you, our clients, towards a successful understanding of metabolite identification needs in support of drug development requirements. Fred Pritchard is Celerion's Vice President of Global Drug Development. Dr. Pritchard brings over 30 years of drug development experience, including pre-registration work on 5 currently marketed pharmaceuticals and several that are currently in clinical development. Dr. Pritchard leads a group of consultants and program directors within Celerion, which have collectively worked on nearly 40 integrated drug development programs through IND filing to phase 1 and 2 clinical trials. He is the 2000 Alumni Fellow of the Penn State College of Medicine and was named one of 9 notable people in pharmaceutical R&D by R&D Directions magazine in 2009. Our second speaker is Dr. Sergio Menta, bioanalytical scientist with pharmaceutical chemistry education and 4 years working experience in the drug metabolism and regulated bioanalysis areas within pharmaceutical industry. Sergio holds a doctoral degree in pharmaceutical sciences from Sapienza University of Rome. His core competence is the development of LC-MS methods to quantify and characterize new drug candidates and their metabolites in biological fluids and tissues. Sergio has an excellent understanding of the drug discovery process from the early research phase to the preclinical and clinical development phases. And with that, I turn it over to Fred.
Thank you, Lorraine, for that nice introduction. Today, we are going to talk to you about drug development and the importance of metabolites in the drug development program process. I started my career in a drug metabolism department at a major pharmaceutical company back in the 80s. And thinking back on that experience, I realized that actually the basic questions that we're trying to understand today are much in the same as they were back then. But of course, the difference is in the kinds of technology that we have available today to answer those questions, our understanding of the basic biology and cell biology of the cell that metabolizes drugs, and of course, the expectations now that the regulators have with this expanded knowledge. So today we're going to talk a little bit about why drug metabolism is important as kind of an intro. But most importantly, we're going to focus on what are the expectations of regulators, especially when you are bringing that new drug application or that marketing application to their attention. What are they going to look for in the story of how your drug is being metabolized, and what does that mean? Sergio will finish the talk discussing the various modern methods for identifying and quantifying metabolites. So why do we study drug metabolism? Well, of course, it's an important route of drug elimination from the body. Also, metabolites might be active pharmacologically and contribute to the activity of the parent compound. They may also act at other places in the body, and some of those might not be what you want, and they may be toxic. Furthermore, variations in metabolism among individuals often explains variations in drug response. So drug metabolite profiling is closely connected with our understanding of variations in how the drug is eliminated. And finally, it's important to understand the profiles of drug metabolism in humans and how that relates to the various tox species, because what we try to do is to focus our attention for our toxicology studies on species where the profiles match most closely to humans. Just some basics on drug metabolism. When we talk about metabolism of drugs or other xenobiotics or foreign compounds, we refer to two types. There's a phase 1 metabolism, and this is an oxidation or reduction reaction that changes one of the molecules of the particular drug, and it turns them into more nucleophilic or more electrophilic forms. These can further react with other enzymes that will add larger molecules to the compound in a kind of a conjugation reaction. We call these conjugation reactions phase 2 metabolism, and this includes sulfation by adding a sulfate, acetylation, and glucuronidation. We also have for electrophiles. It's important because these compounds are highly reactive with proteins and sometimes can be damaging to critical elements of the cell. But fortunately, our bodies are equipped with glutathione, which can bind to these highly electrophilic compounds and create a conjugate that can be readily excreted. The whole process is really trying to take these usually highly lipophilic drug compounds and convert them into more hydrophilic compounds that can be more easily excreted by the kidney and in the bile. So an example on the left of this slide is coumarin. It goes actually through this two-stage metabolism process, a phase 1 hydroxylation at the 7 position to make 7-hydroxycoumarin. This in turn is a nice nucleophile for the glucuronal transferases and creates the 7-hydroxycoumarin glucuronide. Now, an active metabolite is one that retains some or all of the pharmacological activity of the parent compound. And here we see an example with imipramine, which is the active drug. And here we have an N-demethylation reaction, which removes the methyl group on the side chain, but it doesn't change the activity of this compound because the activity resides within the ring structure at the top of the molecule. And we have a number of other examples of this. The benzodiazepine family is a classic because almost every oxidative metabolism that happens in this group, the activity is retained. And you can see that there have been several drugs developed out of the metabolites because of that feature. And it's only until you get to a stage where the compound is conjugated that it can be eliminated in the urine. And there are several other examples of active metabolites from active drugs that are well known. Codeine actually gets converted to morphine, and this is an example where the metabolite has actually more activity than the parent drug. So much of the analgesic activity of codeine is due to the conversion to morphine. And the same is with not the same, but digitoxin to digoxin is another example. Amitriptyline and nortriptyline are both active, and procainamide to N-acetylprocainamide are classic examples of active metabolites. So we need to know the identity of these metabolites because they may be active and contributing to the pharmacology of the drug. A couple of other key terms in this field are major metabolites, and generally that's considered to be something that circulates at greater than 25% of the parent drug's systemic exposure, and that means of the parent drug's concentration, or AUC. An important metabolite, particularly under the MIST guidance, which we'll be getting into more, particularly in Sergio's talk, is one that is 10% or greater of the total drug-related material or systemic exposure that you see in the plasma or blood, or that you find in the excreta, like urine or feces. So essentially, a metabolite that accounts for more than 10% of the drug's clearance. And then occasionally we'll find a unique metabolite, which is one that is found in the plasma, or particularly the plasma of only one of the species. That's of the tox species. And this can be an issue, particularly if the unique metabolite is found in humans only. Now, here's an example of the importance of knowing a major metabolite. This happens to be a study where we're looking at the impact of moderate hepatic insufficiency on the pharmacokinetics of a drug. And on the left side, this is represented, the normal situation is represented by the dotted line, and the parent drug is on the left side. And you can see that when you compare that to patients who have moderate hepatic insufficiency, the exposure of the parent drug rises almost twofold by both heightened Cmax and a much higher area under the curve in those patients. So clearly these patients, their clearance, metabolic clearance of this drug is slowed. But because we know that there's a major metabolite, and you can see it here, the major metabolite circulates at almost equal concentrations as the parent drug in normal conditions, but in these patients, it's reduced by 50%. So by having an assay for the major metabolite, knowing what it is, when this study was conducted, we can now say with confidence that in patients with moderate hepatic impairment, they are reducing the formation of this metabolite through this pathway, and we have a very definitive answer as to what's going on in hepatic disease. Now, the key enzymes for metabolizing phase one reactions are shown here, and this is a compilation of all the drugs that we know, how they're metabolized, and what percentage of those drugs utilize these various phase one enzymes. And of course, the big group is the cytochrome P450s, which you can see on this pie chart account for almost 75% of all the frequency of all the drugs that we know of. And the most popular drug, or the most common enzyme that's involved, is the CYP3A system, where it's almost like 40% of the enzyme of drugs that are metabolized by phase one utilize this enzyme. But close on its heels is CYP2D6 and CYP2C9. And what's significant about these two, 2D6 and 2C9, is they are polymorphic. That means they have functional alleles at the gene level that are capable of producing people who do not have good capability of metabolizing the substrates that normally this enzyme would use. So you have poor metabolizers in these two classes of enzymes, and some others as well. And this has consequences in explaining the variability in drug metabolism and hence drug response. And the most common forms of drug metabolizing enzymes for the phase two reactions are the glucuronidal transferases, which you see here is also about 35 or 40%, and the sulfyltransferases, which is another 20 to 25%. And there are some glutathione S transferases as well, and some other various conjugating enzymes. Some of them, of course, are normal cellular enzymes that are that are involved in normal cellular metabolism. So we're kind of hijacking those enzymes for the drug. And a good example are the thiopurine methyltransferases and the catechol O methyltransferases. But they are fairly rare enzymes involved when you look at the big picture. What's important about the phase 2 enzymes, even though the products of these are usually excreted, is that most of them are polymorphic as well. So this can contribute to great variations in drug response if this is a pathway that's important, especially for the parent drug's elimination. So the key questions that we need to know about drug metabolism, certainly at the time of NDA submission, are these. What contribution does metabolism make to drug clearance? And we get a lot of information about this from PK studies, but ADME studies are the prime source for this for both humans and our tox species. What human drug metabolizing enzymes are involved in the metabolism of a drug? Nowadays, we can get this information well before entering clinical trials because we can incubate purified preparations of these human enzymes with our drug and see if that enzyme will metabolize that drug, or if that drug is a substrate for the enzyme, another way of saying it. We can also use various liver preparations from various animal species and humans to do the same thing, and then we can also but also later in clinical development, as we're doing the drug-drug interaction studies, we're building on our knowledge of what the pathways of metabolism are doing, and by knowing more about which inhibitors matter will tell us a lot about which pathways are the most important in a drug's metabolism. And finally, understanding the poor metabolizers. If you have polymorphic human drug metabolizing enzymes involved in your drug's metabolism, this can add additional information about the importance of the pathways. And then finally, the big question: what metabolites are produced, and how do we identify those? That's what Sergio is going to talk about. But we need to compare these profiles, as I said before, between human and the various tox species used. And we also need to identify those unique metabolites. And then, knowing what the metabolites are, we need to determine if they have any pharmacological activity. So that's what the regulators are looking for, basically, when they're reviewing the NDA, the accumulation of knowledge that we have that tells that story. So the ADME study in humans is usually done with radiolabel, almost always. And we give the radiolabel and we collect blood samples, which we can split into plasma and red blood cells. And actually, the proportion of drug that stays in the plasma and the red blood cells is important, because that tells you how much of the drug and drug-related material gets into the erythrocytes, and that's a surrogate for how easily that can penetrate into other cells as well. And of course, we collect time collections of urine and feces so that we can capture all the material that's coming out of the body that have where the label of the drug is attached. And so that label will include the parent drug and all metabolites. And so we can do a variety of measures here. The first is we can count, if the middle one, the total radioactivity using scintillation and standard methodology. We can also take these samples and assay them with already validated assays that we've used with cold drug. That would typically be the parent drug and any metabolites assays that we've developed so far. And then the third part is doing metabolite profiling, looking for metabolites that we might not have identified yet, and how important that might be. So this is what we usually get. So if we look at the radioactivity plasma profile, which is the top line under the large arrow observed, the important thing is to remember that that represents not an individual, it represents a summation of all the drug-related material that's in plasma. So we can measure the parent drug, that would be the blue line, and then the various four thinner black lines would be, say, 4 metabolites that have been formed from the parent drug and their time course in the blood. And the observed total radioactivity is the summation of all 4 of those. So that's an important thing to keep in mind. And then, so why is that important? Well, let's take this example. Here we have on the left side a plasma profile. We have the total radioactivity on the top line, and we have the parent drug on the bottom line. And it's pretty apparent that the amount of AUC that's so much greater than what we have with the parent drug means that there's a lot of drug-related material floating around in your body related to the administration of this drug, and we need to characterize what that is for our submission. On the excreted side, we collect these and count the total reactivity in the urine and feces, and this is helpful while the experiment is going on, because we want to know when the amount of excreta is low enough that the subject can leave the site, but also if we've collected enough to characterize how much comes out in the urine and how much in the feces. And in this example, you can see that we've, our target is 90%. If we can recover 90% of what we gave, that's considered a very good experiment. An acceptable level is 80%. And this is the type of data that you will see from that study. And this is an interesting slide created from 23 studies done at Celerion. And it ranks them from amount recovered to, from the highest to the lowest. And you can see that for about half of them, we've recovered 90% of the drug totally. And then as we get a little further, probably for 80 to 90% of them, we've recovered 80%. So most of them, we've hit the target, hit the acceptable range. But there are a few where we had not been able to recover fully the drug. And this can be due to a number of reasons. We do everything we can to make sure it's not a problem with the actual conduct of the study, but usually it's because the drug is being retained in the body for an extended period of time, and we just can't measure that. You also see from this slide that it varies all over the map from drug to drug, how much comes out in the urine, how much comes out in the feces. While on average it's about 50-50, it can go from nothing to 100%. So wide variability across drugs in the whether the total radioactivity comes out urine or feces, and often we don't have a very good idea before doing this experiment of what that balance is. So in doing these studies, there's some key considerations. The first, of course, is the site of the radio label, and that really you need to have the radio label at a place where it won't be cleaved and then lost. It needs to reflect the core of the molecule that that's important for these studies, and also how hot you can make the drug. How high a specific activity can you have with the product, because that will give you a lot more options in administering it to humans or animals. To do a human ADME study, you need a tissue distribution study to do the dosimetry that will determine how much radioactivity you can give to a human being. It's important, these are done in rodents. It's important to at least have some rodents that are pigmented, because melanin in the eye is an important binding site for many drugs. And the eye is the retina of the eye is a radiosensitive target. And it's an important consideration in these dosimetry studies. You do the calculations, and we can certainly help clients with that. And then you prepare the radiolabeled dose, And that's usually involved a dilution of the radiolabeled drug with cold drug, usually 100 to 1 or more. And but there is an option with microtracer that we can talk to you about further. The problem with microtracer is it's such small doses of radioactivity that you need a very advanced accelerator mass spectrometer to do the analysis. So the analysis can become quite pricey, but it is an option for certain drugs. The duration of study is important because you need to keep the participants in the study in a very confined area until you've either collected your 90% of the drug or they're excreting such a low amount of radioactivity it's not going to be a hazard to the community. And then finally, the stability of the samples, how you process them and get them ready to hand off to the lab that's going to do the metabolite quantification and identification work. And with that, I'm going to hand over to Sergio, who's going to tell us what he does with the samples and why that's important once they are received in the lab. Sergio?
Yes, thank you, Fred. Thank you for this introduction. And as you already mentioned, so due to the possible contribution of drug metabolites to the pharmacology and toxicology of new drugs, the drug metabolism studies are crucial during drug development in order to ensure the safety and efficacy of new therapies. And the main guidance addressing regulatory requirements in terms of drug metabolites is for sure the Metabolites in Safety Testing from FDA, which was first issued in 2008. And due to its introduction and also thanks to technological advances, methods for metabolites identification and quantification have changed. And what the guidance says about human metabolites can be mainly included in the last two bullet points. So I will also discuss them later in further details. So actually, human metabolites exceeding the 10% of the area under the curve at steady state are those that require further evaluation in a way that it's important for these metabolites that at least one preclinical toxicology species has to show greater steady state exposure to these human metabolites. But what did it change with the introduction of the MIST guidance? Actually, before the MIST guidance was issued, radiochromatography was the method of choice to generate fully quantitative metabolite profiles already in preclinical species and generally in preclinical development after in vitro administration to animals on human or in life administration to animals of radio-labeled compounds. While the study of human samples was generally delayed and performed in the context of the human radio-labeled ADME study, which was performed in general at the end of phase 2 or during phase 3. But then after the MIST guidance was introduced, the key point was an increased focus on human metabolites and their potential contribution to safety and drug-drug interactions. And this means that the generation of human quantitative metabolite profiles became more urgent in drug development. And due to this reason, today the conduction of the human radio-labeled ADME studies generally performed during middle of phase 2, and also new technologies are employed to determine preliminary metabolite profiles already during phase 1. Altogether, this led to a reduction of animal radio-labeled studies during preclinical development. And a typical strategy that is followed by drug metabolism scientists to address the MIST guidance can be divided in 3 stages. So for sure, the stage one is during preclinical development when compounds are incubated with in vitro in order to generate and to identify metabolites, which are then compared across animals and humans in order to select the suitable animal species to perform the in vivo toxicology experiments on the basis of metabolic similarities with respect to human. And also in vivo animal plasma profiles are used in order to understand the predictive power of the in vitro models and also to establish potential in vitro in vivo correlations. And at this stage, the comparison across species is mainly based on chromatographic retention time and mass spectrometry data, since, as I already anticipated, the misguidance led to a reduction of radiolabel studies in preclinical development. A typical metabolite identification workflow starts with the in vitro incubation of a test compound, generally in liver microsomes or hepatocytes from multiple species in our bioanalytical labs, or with the in-life administration of the drug at the animal facility. And this is followed by sample collection at different time points. Then the second step is the sample cleanup, in which we develop extraction procedures in order to extract the test compound and all its metabolites from the matrices. And then we analyze extracted samples by means of liquid chromatography, high resolution mass spectrometry, which is a very powerful technique that can be used for metabolite identification. Of course, we face several challenges during method development, because first we need chromatographic methods which are able to retain and discriminate the test compound and all its metabolites, and also we need, of course, specific mass spectrometric workflows which are able to select and fragment as many metabolites as possible in a way that the necessary structural information can be obtained. The last step, of course, is the data analysis. For this, we use MetaID software that, in our case, is the Metabolite Pilot from SciEx, which, of course, helps us in facing the big challenge of metabolite structure identification. Then moving towards the clinic, the stage 2 of this process is during phase 1, indeed, when human plasma samples, generally from multiple ascending dose studies, are analyzed to find out circulating metabolites. And in this context, the use of LC, UV, HRMS techniques is quite powerful, since it allows to get both qualitative data thanks to the high resolution mass spectrometry, but also quantitative data via UV in order to assess relative abundances of metabolites with respect to the parent drug. Besides, as it's impossible at this stage to get fully quantitative metabolite profiles without a radiolabel, and due to the fact that what needs to be demonstrated is that animal preclinical species show higher exposure to human metabolites than human, mixed matrix methods are employed in order to understand if we have the necessary metabolite coverage. In this approach, the human plasma samples at the therapeutic dose are mixed 1-to-1 with blank animal plasma samples, and the other way around. So animal plasma samples at the non-observed adverse effect level are mixed 1-to-1 with blank human plasma samples in order to correct for the different matrix ionization effects. Then samples are analyzed via LC-MS, and peak areas are used to compare the concentrations on a relative fashion between animals and humans in order to understand whether the toxicology species show a higher amount of human metabolites than human, or in other words, to understand whether there is the necessary coverage that is needed for under a regulatory perspective. Also, additional approaches include the use of the animal radiolabel studies to calculate the radiometric response of human metabolites, so that to correct the mass spectrometric, the different mass spectrometric response, in order to quantify also human metabolites from non-radiolabeled human samples. And of course, NMR spectroscopy has been widely used for structure refinements, and also is used for metabolite quantification. And last but not least, the accelerator mass spectrometry is an emerging technology that allows the conduction of microtracer ADME studies, in which actually a much lower amount of radioactivity can be administered to patients. Then the last part, so the last stage of this procedure is during phase 2, and by the end of this phase, all the necessary metabolite assessments have to be completed before starting large phase 3 clinical trials. And this is the time in which the human radiolabel ADME study has to be conducted, since it provides the pharmacokinetics and fate of total drug-related material together with fully quantitative metabolite profiles. And by means of this study, it's really easy to understand which are those human metabolites that are exceeding this 10% threshold of the total drug-related material at steady state, in order to address the missed guidance. Because what the guidance says about this disproportionate metabolites can be summarized in this flow diagram. And if we start from the left, so human metabolites accounting for less than 10% of the total drug-related exposure at steady state, they don't require further evaluation. But human metabolites accounting for more than 10%, so now on the right, are those that can raise a safety concern, and for this reason they require further evaluation. And in this case, the first question is whether they were also observed in any of the preclinical toxicology species or not. If the answer is no, of course, the synthesis and non-clinical testing with the drug metabolite is required. While if the answer is yes, so if this human metabolite was also observed in at least one preclinical species, then it's necessary to look at exposure. And in this case, if the animal exposure was lower than the human exposure, again, the preclinical testing of with the drug metabolite is required, while if the animal exposure to the human metabolite was higher than the human exposure to the same metabolite, of course, no further testing of the drug metabolites are needed. A typical human ADME study at Celerion starts in our clinic with the administration of a radiolabeled compound, usually with C14, to 6 to 8 individuals, and with subsequent sample collections and different time points. And samples are then analyzed via liquid scintillation counting, and until the total amount of radioactivity can be recovered in excreta. Then samples are, for MetaID and profiling purposes, are shipped to our facility in Zurich, where what we do, we develop sample pooling procedures in order to address the misguidance. So we pool samples across time points or across individuals. And then after sample pooling, we develop, the second point, we develop extraction procedures in order to recover the maximum amount of radioactivity from each sample. After sample extraction, we analyze the extracted samples by means of liquid chromatography, accurate radioisotope counting, high-resolution mass spectrometry, which is a cutting-edge technique that allows to get both qualitative and quantitative data. Indeed, we can, of course, elucidate metabolite structures by means of high-resolution mass spectrometry, and also we can quantify each metabolite on the basis of its radiometric response by means of online radiodetector method. And in general, what we do, we characterize all human metabolites exceeding this 10% of the total drug-related material steady state in plasma, and also exceeding 10% of the dose in excreta. But every detail is always discussed with the sponsor. It depends on needs and timelines, of course. Here, just some our LCMS capabilities at Celerion, Switzerland. We have several qualified scientists with more than 20 years experience in metabolite identification. We have triple TOF 6600 plus from high resolution mass spectrometers from SciEx, together with the BRC3 radioisotope detector, and also, of course, triple highly sensitive triple quadruple 6500 plus for targeted quantitative analysis, together with UPLC systems from Waters and SciEx. And then also, of course, we use latest software packages from SciEx for metabolite identification and quantification. We also have 21 CFR Part 11 compliant LIMS, electronic laboratory notebook, and temperature monitoring systems. Our labs are GLP GSCP certified and inspected from Swissmedic and FDA. And also we have state-of-the-art cell culture and laboratory equipment. And with this, I'm done, and I thank you for your attention.
Thank you so much, Sergio. That was a really informative session. I'd like to thank both Fred and Sergio, and we're going to now move to the Q&A period. And Fred will address the questions that have come in for your presentation first, followed by Sergio's. So I'm going to go through those questions now. And Fred, the first question we'd like to take a look at is, when is the ideal time during the early development program to plan a human metabolite identification study? And how is it best to pre-plan during the early first-in-human studies to include this work?
Well, as Sergio mentioned, in the old days, people used to leave this type of study till near the end of drug development, kind of to complete the NDA. But with the misguidance, it's important now to determine what you have earlier in clinical development. So ideally, it would be good to do a human radiolabeled study sometime in phase 2. When you know that your compound is progressing in efficacy and dose response trials, and that you're really preparing for those full efficacy trials for phase 3. You'll want to know what are the major metabolites. Do we have tox coverage where there's time, while there's still time to adjust your tox program, chronic tox program accordingly. And so that would be the ideal time. Now, the other advantage we have today, and Sergio mentioned, is that we can get quite a bit of information from the multiple ascending dose study, from both the plasma and occasional urine or samples there, as to what might be the major metabolites using cold methods. And that can give you a little bit of a jump start to help plan that phase 2 study and know what's important. It does take about 6 to 8 months of lead time to get your synthesis of a radiolabel down, and also to do the rodent tissue distribution studies and do the dosimetry calculations before actually executing your radiolabeled study. So this is really activity that needs to be begun while the latter part of phase 1 is happening typically, and certainly plan for as you begin to enter your clinical study program.
Great. And in a related question, we had briefly mentioned toxicology. Maybe you could comment on what the regulatory approach might be when your toxicology species and the human species do not have a strong metabolic profile similarity.
Yeah, that happens from time to time, and the typical species for rodent, mice and rats, and non-rodent are dogs and monkey. There are some other species that could be investigated, and that's why sometimes it's important to get this information as early about the human as early as you can. But it does present a complication for both the reviewers and for the sponsors if the human is somewhat unique to the tox species. And that does happen, and it requires a fair amount of conversation with the FDA as you move forward to really resolve that. So that the solutions could be a somewhat uniquer species in the tox program, but of course the problems with that is you don't have a long history of how those species do in tox studies. So it's a trade-off, and it requires discussion with the agent, whatever regulatory body you're submitting to.
That makes sense. And the other question that everyone wants to know is it possible to submit a successful regulatory submission without ever doing a human radio-labeled ADME study?
Yeah, that we get that question a lot, and I think today it's probably more possible than it ever has been. Obviously, if you have a large molecule drug that or a peptide that breaks down into normal body components, you don't, and if you can demonstrate that, that it's theoretical, certainly for peptides or oligonucleotides that are not somewhat substituted, that you don't need to do a human ADME in those situations. Also, if you can demonstrate through cold methods that say all your drug is, or 90% of your drug is excreted in the urine, and or as parent drug, then it's not being metabolized much. So you don't have so if you can demonstrate that your metabolic clearance is less than 10% of total clearance, that would be another reason for not having to do a radiolabeled study, especially if you can quantify how much is coming out in the urine. And you may get lucky where in the early phase, you may have a very simple profile of metabolism where you have 1 or 2 metabolites that you can easily measure using cold methods, and through that quantify 90%, 80 to 90% of your ADME without reverting to a radio label. So it's possible. It's not common, but it's something that you can assess whether that's the case early in phase 1 drug development.
It's a lot to consider, and it really reiterates the concept of pre-planning and a thoughtful approach. So thank you very much, Fred. I'm going to move our questions over to Sergio's presentation. And Sergio, maybe you could comment on the common pooling strategies for metabolite profiling purposes.
Yes. I mean, in general, what we do in order to address the guidance, we prepare an AUC plasma pools across time points for each individuals in order to estimate metabolite exposure at steady state. And also the same type of pool is prepared for excreta, so basically across time intervals, in order to identify the measure excreted metabolites. Another pooling strategies is indeed across individuals per time point, and this pooling strategy allows to extrapolate the PK profile of the test compound and all its metabolites. So these are the 2 main pooling strategies, basically across time points for each individual and across individuals for each time point mainly.
Right. Now, as you have those samples, maybe you can also add in a conversation around the advantages of using a high-res mass spec for these identification studies.
Yes, actually, high-resolution mass spectrometry is from this point of view, is a very powerful technique because it allows to measure the exact mass of a compound, and of course, this allows to distinguish even minor changes in molecular structures, and which is the case of metabolites. And for this reason, this is extremely useful. It's a useful technique for the conduction of metabolite identification studies.
One final question. You had mentioned a mixed matrix approach. Maybe you could comment on the reliability of this approach for phase 1 to assess the metabolite coverage across the species?
Yes, so actually on the basis of statistical data, they show that when everything is based on the LC-MS peak area, and when the LC-MS peak area ratio between animal and human plasma samples is more than 2, we can say that it's practically safe to conclude that animal species has an exposure to the human metabolite that is at least equal to or greater than the human exposure to this metabolite, with a confidence about 99%.
That's impressive. Thank you, Sergio. Well, both Fred and Sergio have given us a lot to think about today, and we'd like to thank them for their time. Thank you, everyone online, and this concludes our webinar. A copy of this webinar will be available on our website, and as a reminder, Celerion will address all remaining questions directly within 24 to 48 hours. Thank you once again.